So unfortunately, David Krujai could not be here. He had an emergency, so it is my pleasure instead to introduce Dr. Simon John. Dr. John is the Robert L. Burch III Professor of Ophthalmic Sciences at Columbia University's College of Physicians and Surgeons. He earned his PhD in biology and human genetics at McGill University in 1992 and trained under the human geneticists Brisma Rosen, Charles Scriver, as well as the Nobel laureate Oliver Smithies. He then went to the Jackson Laboratory in Maine in 1995, where he pioneered the use of mice for glaucoma research by adapting tools from the human clinics, as well as developing new tools and mouse models to study this blinding disease. In 2019, he moved his lab to the Department of Ophthalmology at Columbia University, and that's where they continue to work on the role of the metabolism in glaucoma pathology, which he is going to talk to us about today. And with that, Simon, if you want to take it away.
Thank you, Fiona. Good morning. Just a warning for everybody. I was told there were different audiences for the talks today, and so my talks are largely overlapping, so it's probably not worth going to both. What I'm going to introduce to you today, expecting primarily residents this morning, is how we've been developing resilience-based therapeutic strategies for glaucoma. So first, I'm going to introduce the resilience concept to you. Then I'll go through some of our work that led us to this concept and talk a little bit about how it might be applied. So the resilience concept is really quite simple. It's the use of metabolism supporting or anti-stress treatments to enhance survival, function, and/or regeneration of tissues and cells. And this can be applied in regeneration settings or in age-related settings. One way to think about this is giving the body a tonic. If you're giving it a tonic to keep everything healthy and build cellular resources, those cells are going to be healthy. So the idea is that with glaucoma and/or stresses of aging, there are various deficiencies in metabolism, oxidative stress, and other damaging processes which are damaging vision, damaging the cells with age. If we can support metabolism, we basically increase cellular energetics, increase cellular resources and cellular function, because the cell has more energy and resources to deal with stress, to repair stress, to protect itself, etc. And this induces a resilience state which counteracts aging and disease. So you might have a much healthier aging and fend off glaucoma. So that's the basic concept of resilience. The resilience can be neuroprotective, which is where we've demonstrated most of it so far. But also, as I come to the end of the talk, I'm going to show you some cases where applying it to intraocular pressure itself, because really it could be applicable to any tissue in the body. These treatments are anti-aging, anti-disease, pro-survival, pro-regeneration. Why are we interested in resilience? Well, if you target a specific gene or a specific molecule of the drug, it's got limited numbers of patients it can benefit. Resilience is generally gene and molecule agnostic. If they confirm resilience against aging and general stresses, you could help a broad swath of people. So it's more generally applicable than target or gene-specific approaches. My lab is very interested in using nutrients and metabolite-based approaches, and they have great potential. Other methods include gene therapies or differential resilience strategies others are developing as well. So I'm sure you're all well aware of glaucoma. It's a major cause of blindness affecting 80 million people. These are complex diseases that have in common the death of retinal ganglion cells and the degeneration of the optic nerve. As you know, primary risk factors for glaucoma are genetics and family history, increase in age, and harmfully high intraocular pressure. And the greater the exposure to intraocular pressure you have, the longer, the higher the duration, or possibly the most fluctuation, the worse the disease can be. And so treatments to lower IOP to a safe level are the standard of practice. But they don't work in everybody as adequately well as we would like. higher intraocular pressure is the key risk factor for glaucoma. And in studying intraocular pressure, we have to think about the trabecular meshwork resistor in the iridocorneal angle and the Schlemm's canal. The biology of neither of these tissues is particularly well understood. Despite many studies, there's a lot left to learn. How does the trabecular meshwork function? How does it signal the Schlemm's canal? Exactly what are all of the molecular mechanisms governing a fluid crosses this inner wall into the canal? And how can we keep these tissues healthy with age? Because there is some evidence of demise due to oxidative stress and other problems, of fibrosis, cell death, etc., with age and during glaucoma. So my lab, to study decreased drainage and RGC death, we've traditionally used a lot of inherited models to get at the primary causes of disease. We've also used inducible models to study the retinal ganglion cell death. In using these models, we've used the full array of genetic approaches and gene mapping identification and genomic approaches, meaning you're studying all of the genes across the genome or how the genome works to study these diseases. A lot of our recent expression has been using gene expression profiling across the genome, a form of genomics, to identify very early stages of disease. And we've been moving into single cell and spatial profiling to try to understand both the drainage loss and the retinal ganglion cell death. I won't go through other things on this slide now, but the rest of my talk is all growing out of these modern contemporary genomic gene expression types of studies. We're doing a lot of work. We've recently published a few important papers, or they're on BioRx under review, using single cell resolution genomics to transform understanding of the functions of these tissues. Other groups are also doing this work. So there's a lot of information being learned recently about these tissues. These technologies are very important, these single cell resolution technologies, and they're very important because they allow you to analyze the transcriptome of individual cells. This allows understanding of heterogeneity of cellular responses and cellular roles in disease types. And we know what's happening in individual cells. So that's very important. In our previous methods, if you were looking at RNA or gene expression, you were averaging the results of a whole tissue, which might not only didn't look at individual cells of a given cell type, but it also averaged across all of the cells in the tissue. So it wasn't anywhere near as powerful. These techniques do have these limitations of sampling, but nevertheless, they are still transformational and are teaching us a lot about various aspects of this part of the eye, including development, the cellular identities, the transcriptional regulation, and the roles of different cell types. And by studying mutants, and I'll give you an example at the end, we can also start to understand disease initiation and progression. We've recently either posted or published papers for the Schlemm's canal and the trabecular meshwork. For Schlemm's canal, we defined 3 transcriptional states of cells, including outer wall and 2 inner wall cell states. We've also, over the years, shown that these cells of the Schlemm's are molecularly both vascular and lymphatic, they're in between. And we've clarified some errors in the literature and have shown that they're clearly more lymphatic biased than vascular biased. For the trabecular meshwork, and I want you to remember this for the end of the talk, please, we've defined 3 subtypes of cells in mice, TM1, TM2, and TM3. Various groups have reported different numbers of trabecular meshwork subtypes. We believe that some of these were over-clustering, and not all of these cell types are proven reliable. We collaborated with Dan Stamer's group at Duke and with Vati Balasubramanian, who's talking in Utah next week. Was a senior person in my lab, and I was our own lab, did a lot of this work along with Nicholas Tolman in my lab. And in collaboration with Dan, they were able to show that these cell types are very robust across institutions. We used data from Duke and Columbia and found these cell types. So we've got great confidence in the cell types that we're reporting. We can't say yet we've identified all of the subtypes, but we have identified these 3 important ones. Importantly, these cell types, although they're clearly TM cells and they have much overlap in the pathways that are active in them, They also have unique enrichment for specific pathways, which differs between these subtypes. For example, the TM3 subtype is enriched in metabolic processes. And here you see a UMAP on the right showing some of these subtypes we got from mice, the TM1, 2, and 3 in the pale blue, red, and green, occupying this dimensional space. And if you look at them by heat maps of the top 15 to 20 signature genes, you can see they're clearly distinct from each other. Each of these little hashes is a single gene. Coming across this way, they're different cells. On a single sample, a single cell coming this way. So you can see all of these cells clustered into TM3, and in this case, the genes that are the most yellow were the highest expressed, and the ones that are darker were the lowest expressed. We've also been working on the development of the drainage ankle throughout its stages of development postnatally in mice. This is very important for understanding childhood and adult glaucomas. And it's leading us in extending our resilience approach to believe that dietary interventions could become an important paradigm in the treatment of developmental glaucomas, and I'll touch on that very briefly at the end of the talk. So to move on, today what I'm going to focus on, because Arthur will be talking about a lot of the single cell work next week, is how we're moving towards resilience boosting strategies against intraocular pressure elevation and glaucoma. We initially discovered metabolic disturbances within retinal ganglion cells that contribute to neurodegeneration in mice, and we're finding that resilience strategies are important against those processes. And the work at the end of the talk is more recent unpublished data, which demonstrates a role for metabolism and resilience treatments in countering IOP elevation. I'm going to start by telling you about some neurobiology experiments we did with DBA2J mice. This is an age-dependent model of inherited glaucoma, and using these mice, we uncovered metabolic defects in glaucoma initially, which have now been confirmed in other species and have led to clinical trials around the world with initial positive results in 2. These mice develop a pigmentary form of glaucoma resulting from damage to the iris. The iris disease is initiated by genes affecting melanosomal pathways. For example, this GPNMB gene, glycosylated protein NMB, and tyrosis-related protein 1. We showed that this induces iris disease through a toxic melanogenic process where toxic metabolites leak out into the cell from melanosoma and damage iris cells. This is now the same in at least a subset of human PDS or pigmentary glaucoma, because the PMEL70 gene is a melanosomal gene that's been implicated in that disease by Michael Walter's group. Also, in these mice, we see identical melanosomal abnormalities, or essentially identical to those that have been reported in some human patients a long time ago. So there's molecular overlap here in the initiating causes of disease, even though the pigment dispersion on iris disease in these mice is a lot more severe than you see in a typical person, because you've got these 2 mutant genes that conspire to cause a very severe iris disease. This iris disease and the pigment dispersion results in pigment being deposited into the drain, which elevates intraocular pressure and causes glaucoma. Regarding the timeframes of disease, the iris disease is kicking in floridly by 5 to 6 months of age, with increased intraocular pressure in most animals by 6 to 8 months, and retinal ganglion cell death and optic nerve atrophy by 10 to 12 months. Importantly, we're able to derive mice with a wild-type version of the GPNMB gene. We got them out of the freezer from 1983. We'd worked out this mutation occurred around that period of time. And we bred this mutation to modern-day DBA2J mice. We've got genetically matched control mice, and these mice do not develop high intraocular pressure or glaucoma. So they're behind many of my experiments, even if, or my lab's experiments, even if I don't mention them. This shows you typical glaucoma in DBA2J mice. I can see the nerve fiber layer is thinned out, and this nerve is excavated, as you might have in a person. But the disease in our colony is really pretty specific to retinal ganglion cells. There's thinning of the nerve fiber layer and RGC loss, but the rest of the retina is intact. So using DBA2J mice, we wanted to ask how the neuron is insulted. What are the early molecular changes that precede axon and RGC loss in glaucoma? processes that are damaging occur in both the retina and the optic nerve. Our work has shown some independence between these, but has also suggested a critical insult in the optic nerve head, as has been suggested in human and other glaucomas. And we actually experimentally demonstrated that in DBA2J mice. So it's actually the only model in which a direct axon insult within the optic nerve has been directly demonstrated experimentally. Using these mice, we decided to use genome-wide expression studies, and we designed these studies to identify the very earliest stages of damage or molecular changes that precede any neurodegeneration. So the experiments I'm going to tell you about, please remember that before any neurodegeneration. We've also done cell type specific studies, which I'll come to, and we've also been interested in what increases neuronal vulnerability with age. How did we design studies to get to the very earliest stages? Well, we could take these control mice that we had and house them in the same cages. So these are mice that differ with a single point mutation, but are otherwise genetically matched to the mice that develop glaucoma, but these mice do not develop high intraocular pressure glaucoma. And these mice, then as we age them, are age, sex, and environment, and everything else matched to the DBA2J mice that go on to develop glaucoma. Now, by studying mice at an age where, excuse me, they had a mix of damage severities. We had mice with very obvious damage, and what we'd call moderate 30 or more % axon loss, or very severe damage greater than 50% axon loss, and a whole bunch of mice that weren't distinguishable from each other morphologically or physiologically. And these mice, we reasoned that using these molecular gene expression approaches, we'd be able to separate them into different groups. So what we did is we took all of these mice, analyzed their gene expression, and then computationally analyzed this data using a technique known as hierarchical clustering. And we did this in a non-biased way. So the computer separated the mice into groups based on the relatedness of each sample across all of the expressed genes that were glaucoma relevant, and it recreated this control group. It recreated our moderate and severe groups, and it actually separated these previously indistinguishable groups from control into 3 stages, which had increase in disease severity. Having succeeded there and tested some of the mechanisms very early in the disease, we moved into experiments specifically within retinal ganglion cells, and using the same techniques, we were able to subdivide retinal ganglion cells from mice that had no detectable glaucoma by conventional criteria into 4 groups that preceded any damage. And just remember that all of these eyes had ongoing high intraocular pressure but hadn't yet developed glaucoma. Also, please remember that even though group 4 might sound late, it's an early stage of disease because it's prior to neurodegeneration. I'll show you some of the data. When we did this, we sequenced 82 separate samples with about 25,000 cells per sample. And when we clustered these eyes, many of them, because we used a reasonably young age, clustered with our controls. So these were related to each other and not significantly different from each other. And here we had our GPN and B no glaucoma controls at either a young age, 4 months, or an older age, 9 months. And we had some of the DBA2J mice at the same ages clustering in the same way. So all of the mice at 4 or the controls at 9 months didn't have any glaucoma, and a subset of the DBA2J mice at the same age did not have any glaucoma and clustered together. Group 2 was the first cluster which was statistically significant from glaucoma, and you see it's got a small number of differentially expressed genes here compared to the controls, and groups 3 and 4 were later stages of glaucoma as shown here. Excuse me. Using this method, we discovered that mitochondrial dysfunction occurs very, very early in glaucoma. If we look at our D2 control mice, the GPN and B mice that don't develop glaucoma versus our group 1, they answer differently different in the ratio of mitochondrial to nuclear gene reads. However, by the time you get to groups 2, 3, and 4, there are substantial differences here. And this suggests that the ratio of mitochondrial transcripts to nuclear transcripts, it's shown us that it's wrong, supporting mitochondrial dysfunction. If we look at the pathways that impact mitochondrial function and metabolism, as early as this very earliest molecularly defined stage of glaucoma, group 2, we're seeing significant changes, such as in the EIF2 pathway, which can control oxidative ER stress, but also is very important for mitochondrial health. If we look at mitochondrial dysfunction pathways, they're changed very early. If we look at other metabolic pathways that regulate metabolism, changed early. And if we look within mitochondria at oxidative phosphorylation, the process by which you make ATP, there's abnormalities in this pathway very early on. If we look at this as a heat map, we have our GPN and B controls. We have our 4 stages of early glaucoma in retinal ganglion cells. We can see how group 1 doesn't really differ to the controls. By the time we get to groups 2, 3, and 4, there are substantial differences. Again, these are individual genes, each little rectangle. They all belong to all of these complexes of the electron transport chain. And the redder the color, the more highly expressed, and the blue, the more lowly expressed. So we had substantial differences with the ETC. We also detected differences in mitochondrial morphology, as shown here in the controls, the mitochondria have cristae, and in some of the DP2, some of the mitochondria lose their cristae and look abnormal. So this all supported mitochondrial dysfunction, and this led us to want to study metabolism. So we did some metabolomics. Pete Williams was very important in this work when he was in the lab. And so we did metabolomics in collaboration with Craig Whitlock, and we also did targeted looking at specific pathways. And what we uncovered, our metabolic profiling uncovered reduced NAD levels and reduced glutathione levels, which changed due to age and disease. This is consistent with altered energy metabolism and increased susceptibility to oxidative stress in these mice as they age and as they develop disease. Here you see in the DBA2J mice, the retinal glutathione levels decrease. Here you see retinal NAD levels decreasing. And here I'm showing you the control GPN and B mice. And because it differs with age in these animals, some of these changes are not due to the glaucoma itself, but due to age. And then when you add glaucoma on top of it, it gets out of control by adding elevated intraocular pressure. In the optic nerve here, again, you can see decrease in NAD levels with age in these mice that are developing glaucoma. So seeing this decrease in NAD levels with age, and that mitochondrial dysfunction gets worse when you've got age and high intraocular pressure, we generated the hypothesis that age-dependent decline of NAD is key in neuronal vulnerability, and that by having lower NAD levels, you increase the probability of metabolic failure when retinal ganglion cells are exposed to a high intraocular pressure and all of the stresses that induces. Now, if this were true, then increase in NAD levels should decrease the likelihood of glaucoma by lowering the likelihood of neuronal failure and the degeneration. So we decided to test this by using nicotinamide. Nicotinamide is the amide form of vitamin B3, thought to be very safe, so it could really have made a human treatment if it's effective. So that's why we decided to test it. Nicotinamide is converted by the enzyme NAMPT, nicotinamide adenine mononucleotide phosphorylase transferase. Excuse me for getting my words mixed up here. And this is converted in the nicotinamide mononucleotide, which is then converted by NMNATs into NAD. This is known as the salvage pathway, and it's the major pathway generating NAD in the body. So what we found is that dietary administration of nicotinamide profoundly protects from glaucoma. It increased NAD levels. It prevented the loss of pattern ERG. It prevents loss of axon transport. It prevents mitochondrial abnormalities, and it protects retinal ganglion cells from optic nerve degeneration. I'm only showing you a subset of the data in the interest of time. But here you can see if we look at flat-mounted retinas in the control stained with RPBMS, we can see lots of retinal ganglion cells, and there's no optic nerve degeneration. You have nice, healthy axons here. In an animal that hasn't been protected and has developed glaucoma, you see a loss of most of the cells, of the ganglion cells, and really a glial scar remaining. And in nicotinamide-treated eyes that are protected, you see preponderance of retinal ganglion cells and a healthy appearing optic nerve. Now, I don't want to show you a few sections. As I mentioned earlier, any age, as you can see here, here's a control untreated animal. Some of them have more detectable glaucoma by conventional means, but some of those might have those earlier molecular stages I showed you, so we call them no or early. But by any traditional measure, they do not have glaucoma. We have a subset of the animals with a moderate stage of glaucoma, and then a subset with severe glaucoma at this age. Because of this variability, you really can't be doing neuroprotection experiments with 4, 10, or 20 animals. You have to use much more robust animal numbers, and we're typically using 60 to 100 eyes in any group that I show you. And that allows us to have robust results with such a variable disease. In using these sample sizes here, we've got our food water control and our food control. We needed both because when we tested different doses, we couldn't put it all in the water, so we had to mix it between both methods. When we give nicotinamide at an early age, it is profoundly protective from the glaucoma. Remember the mice in green, you just really need to focus on them. They don't have glaucoma. It's a substantial increase over the controls. If we started it a little bit later at a time when IOP was already elevated, we also had a profound protection. I don't believe they were more protected at this age. I believe that's just flux of the mean. So maybe the real value is somewhere in between here. And if we give them a higher dose, then 93% of the animals did not develop glaucoma. So this was really a very profound rescue and very encouraging. Overall, the high dose lowered the risk of developing glaucoma by tenfold. This was a remarkable result. So how do these doses relate to what you might want to use in a human? There have been various studies using high doses, often 9 grams or even 20 grams a day in some cases. And in these, there have been very rare side effects, and primarily when you're using high doses of 9 grams or more. When we started our experiments, it is generally believed that 3 grams per day would be safe, but that higher doses may have a toxic potential. The dose, our low dose I showed you is less than 3 grams per day. It is about 2.7 grams per day equivalent for a human. Our high dose, however, was about 9.8 grams per day for a human, so we didn't want to treat people with that. So we started when we went to human trials to use this 2.7 gram per day dose equivalent, so we just rounded it up to 3 grams per day. In doing that, it's been largely safe, but a few adverse events have been seen, and so we do have to revisit that, and patients do have to have liver function tests, etc., to be on a dose as high as 3 grams per day. But generally, overall, they're reasonably safe, and there's a good safety potential. So having discovered, excuse me, that metabolic disturbances contribute to the vulnerability of glaucoma, we wanted to ask, are there other metabolic processes beyond those affecting NAD levels? Can we detect any other metabolic processes that might be important? Indeed, we did further RNA-seq and looked at our samples in more detail, and we found that across these stages in the retinal ganglion cells, we also detected abnormalities in pyruvate metabolism and glucose metabolism, so more generalized metabolic defects than we initially understood. If we look at individual genes, we can see mitochondrial pyruvate carriers, monocarboxylate transporters, etc., changing. Why is this important? Well, pyruvate is a key product of glycolysis. It's also used in a variety of anabolic and catabolic pathways and supports the tricarboxylic acid cycle. So having disturbances in these pathways could have profound consequences. So we wanted to go on and test, does decreased glycolysis affect DBA2J glaucoma? And does pyruvate protect? When we did metabolomics, again with Pete and Craig's labs, we found greatly increased glucose levels and decreased pyruvate levels in the retina of the DBA2 mice. In this case, the pyruvate levels were not age dependent. They were required an IOP elevation. So this showed us it's one of the interactions of IOP with the retinal ganglion cells in perturbing pyruvate metabolism. Other groups have also found deficiencies in glycolysis in glaucoma, including Denny Simmons and Phil Horner's. Our study in finding the high glucose in the retina and the decreased pyruvate levels was also supporting defects of glycolysis, this important energy production pathway that ganglion cells are quite dependent on. So this led us to believe or to test that pyruvate administration could possibly overcome deficiencies in glycolysis, because obviously pyruvate being the end product of glycolysis could overcome deficiencies in glycolysis. So we administered mice with pyruvate, and indeed we could increase the pyruvate levels. What I also want to mention is pyruvate is converted into acetyl-CoA, and this is then converted into citrate as part of the tricarboxylic acid cycle. And low citrate levels had been reported in glaucoma patients, adding relevance. So when we gave dietary pyruvate, we actually prevented glaucoma quite significantly again in the DBA2 mice. And here you see that here's our untreated animals in this experiment. In this age, where a little over 25% have no glaucoma. When you give pyruvate, that's up to 50% or more. And if we give nicotinamide, it was a little bit more, but not too different. And when we give both, even though it's hard to improve this effect, we increased the effect further. And this is all using our lower dose of nicotinamide. If you look at it just in terms of the eyes in green, which have no glaucoma, you can see our water control had the most, with glaucoma, sorry, our water control had the most glaucoma, and then it decreased across these doses with a significant interaction. So combination treatments using nicotinamide and pyruvate are the most effective in our hands. Now, in addition to affecting metabolism, pyruvate can scavenge reactive oxygen species. It induces antioxidant defense by NRF2, and it can promote beneficial autophagy and mitophagy. So like nicotinamide, pyruvate might be having pleiotropic effects. By collaborating with Bob Carson's lab, we also were able to demonstrate that pyruvate protects in a rat-induced model of ocular hypertension, protecting from the neural phenotypes. So this carries across species. So excuse me, I'm going to take a quick drink and then talk about pyruvate a little bit more. Now, I propose that pyruvate is a very important energetic resilience factor, and that's based on the understanding the biochemistry of pyruvate, as well as a collaboration we did with Lou Pasquale. So, as I mentioned previously, pyruvate is a good product of glycolysis. It can then be converted into lactate or interconverted back into pyruvate. Lactate used to be thought of as an anaerobic product, but now it's known to be a fully oxidative product, and it has important signaling roles in metabolism, in modifying other molecules and signaling, etc. So lactate's very important metabolic molecule and very important in cellular signaling, and lactate and pyruvate are interconverted. Pyruvate itself enters the mitochondria via the mitochondrial pyruvate carrier. And in the mitochondria, it's converted into oxaloacetate, which then enters the tricarboxylic acid cycle. And so the tricarboxylic acid cycle is initiated by the condensation of oxaloacetate and acetyl-CoA, which produces citrate and gets this acid cycle, which is also known as the citric acid cycle, rolling. This tricarboxylic acid cycle, as I'll come back to in a minute, is very important in cellular energetics and metabolism. And it's very important in ATP generation because it captures high energy electrons, which then get shuttled into the electron transport chain. So very important for energy production. All of these reasons, and thinking about pyruvate affecting all of this biology, led us to want to look at pyruvate closely. And indeed, working with Lou Pasquale's lab, and these were unbiased experiments, Kiva, Lee, and Lou, they looked at the UK Biobank cohort, And in the UK Biobank cohort with self-reported glaucoma, identified a triad of three metabolites, pyruvate, lactate, and citrate. These three metabolites I just told you about that are very closely related energetic metabolites, with lactate and citrate being interconverted back and forth, and both of these lead into citrate production. Because what I forgot to tell you is lactate can also enter the mitochondria, be converted to pyruvate, and come into the TCA. But in the UK Biobank population, this triad of metabolites was found to confer resilience against the highest polygenic risk scores for glaucoma. And the people in the highest, I think it is quartile of polygenic risk scores, if they had higher lactate, pyruvate, and citrate levels, they were more resilient to glaucoma. And this work was just published in eLife. So this shows very clearly, that nice paper does, that pyruvate and lactate are important resilience factors against human glaucoma, extending the relevance of our work. Now, a little more on the biochemistry. Pyruvate and TCA are foundational in metabolic and biosynthetic balance. Here you see pyruvate coming either through oxalic acid or acetyl-CoA into the TCA. And as an anabolic process, the TCA produces the precursors of most of the major molecular classes in the body, from lipids and fatty acids, nucleotides, amino acids, porphyrin and heme, glucose, and other nucleotides and amino acids. So the TCA is very important for a cell's anabolic processes. However, it's also important for catabolic processes that break down these molecules and generate ATP or energy from glucose oxidation, fatty acid oxidation, lactic oxidation, amino acid and ketone body catabolism, etc. So the TA is central in integrating either catabolic processes which make energy or anabolic processes which make precursors. So lactate and pyruvate are important in regulating this balance, making them very important again. Cells flexibly modulate the TCA depending on need. In some cell states, the primary functions are going to be ATP generation, and in others, through catabolism. In others, they're going to be anabolism. And it's very important that under different set dresses and contexts that the cell gets this balance correct to survive various disease states. So that's why I'm so excited by pyruvate and implicating it and the TCA and metabolism, because it gives us a wonderful target to try to reprogram metabolism in cells, which could synergize with nicotinamide and NAD production. So let me come to intraocular pressure elevation and change gears for a brief time, and then I'll leave time for questions. We've also discovered metabolic changes that are important in IOP elevation, again, using mice we initiated. Our lab has developed an allelic series of new models in the LMX1B gene. We generated these mutations through ethyl nitrose urea mutagenesis, and we identified mutations in different parts of the LMX1B protein. We found these mutations by random screening for glaucoma phenotypes, and it was just chance that, well, not really chance, it was that it was because the LMX1B gene must be so important in glaucoma that in our ENU screen we had this enrichment of LMX1B mutations. We had more mutations by far in LMX1B than any other gene. Mutations in this gene are very important in human glaucoma. They cause the spectrum of human glaucoma from childhood developmental glaucomas all the way to adult onset POG, and have been implicated in human POG via GWAS in individuals who don't have other developmental abnormalities. Mutations in this gene in humans cause glaucoma via intraocular pressure elevation, as most of the studies suggest. There's been some suggestions it could be having direct effects in the retina due to zebrafish studies, but that isn't really supported in mammals, because in all of our gene expression studies, as well as in those we've looked at from others, we've never detected gene expression in the retina. LMX1B encodes a limb domain transcription factor, so it affects the expression of other genes. And what we've uncovered in mice, to cut a long story short, is that the lack of this gene product, or these mutations in this gene rather than lack, cause early mitochondrial dysfunction and cell stress. So LMX1B, also from some work in neurons, is emerging as a stress-inducible transcription factor that regulates stress responses and metabolism. So this is a very important gene and may itself become an important target in our therapies. Looking at mice, early in the talk, I mentioned we'd studied the trabecular meshwork and identified 3 trabecular meshwork cell types, which were very robustly identified, trabecular meshwork cells 1, 2, and 3. I'm going to come back to this TM3 cell type. We found that LMX1B expression is highly enriched in TM3 over other limbal cell types. Also, using single-cell ATAC-seq, this is a method that traps open chromatin regions across the genome. So it's a single-cell genome-wide method for looking at open chromatin, which can allow you to understand active transcription factors by sequencing the regions of the genome that are open chromatin and studying what transcription factor motifs were in them. Using this technique, we discovered that LMX1B is a major transcription factor in these TM3 cells. Excuse me. Further, when we analyzed the TM3 cells in the mutants with this early onset V265D LMX1B mutation, we had discovered metabolic and mitochondrial dysfunction in the mutant TM3 cells, and it was highly enriched in these T3MC cells over any other cell type. This is by single-cell RNA-seq. And you can see that here, we don't have that many cells when you subdivide trabecular meshwork cells down, so the probabilities won't be as high as for if you have a much larger cell set. We only had a total of 2,500 TM cells, and the TM3 cells were a subset of that. But in analyzing the pathways, indeed, it really shows a strong mitochondrial signature, mitochondrial respiratory chain, respiratory complexes, mitochondrial respiration, intermitochondrial proteins, etc. So this showed us mitochondrial changes very early in this model at the stages where the pressure was becoming high, but they didn't yet have glaucoma. This suggested that resilience treatments that support metabolism again would protect against IOP elevation and glaucoma. So we tested this. This shows you the model, and it shows you a protection using nicotinamide. Here you see wild-type mice. In this early onset V265D model, if you don't treat it, you get very deep anterior chambers compared to the wild type, and you see some of the developmental anomalies around the pupil iris here. If we treat them with nicotinamide, we really prevent the anterior chamber deepening, because this is a consequence of high intraocular pressure in these mice. And in mice, anterior chamber deepening is a reasonably sensitive marker of moderately high intraocular pressures. It stretches a lot more easily than it is in human. And because we can't measure intraocular pressure around the clock, it sort of serves as an indelible marker of previous intraocular pressure elevation. To show you that, if you look at anterior chamber depth here, in the untreated wild-type animals that don't have the mutation, there's none. In the untreated, we even have very severe chamber deepening, and most of the animals have deep anterior chambers by 2 months. But the severity and incidence is significantly lower in the treated animals, but it's not completely gone away. We haven't stopped all of the IOP elevation here. Now, these mice, you may say, still have hazy corneas. Well, the corneal phenotype in these mice has 2 components. Part of it is due to high intraocular pressure, but the majority of it is not. The majority of it is due to direct expression of LMX1B in the cornea. It normally turns on collagen genes in the cornea, and when the gene is mutant, corneal collagens, we believe, aren't being made normally, and so you get a hazy cornea that's independent of intraocular pressure as well. Now, if we look at intraocular pressure itself, if we look at our wild-type mice, this is our nicotinamide treated, you can see we've clearly got a lower incidence of intraocular pressure in the treated, which is significant. And even when we go out to 3 months, that change is still there. And these are with robust sample sizes. What about pyruvate? Well, I want to mention different forms of pyruvate quickly. Initially, in our experiments, we use sodium and calcium pyruvate. And in solution, these can be unstable. Now, the way we treat our mice is by putting the pyruvate in the drinking water. So we were concerned about the stability issue. So we've now switched to a molecule known as ethyl pyruvate. Ethyl pyruvate is very similar, but it has an extra ethyl group on it, which does multiple things. It enables it to be transported into cells more easily because it's more lipid permeable. It isn't as dependent on the mitochondrial transporter or the monocarboxylate transporter. So it's more cell penetrant and it's more stable in the drinking water. Now, in mice, we expect a lot of the pyruvate that's making it to the eye through diet of the ethyl pyruvate we put in is actually pyruvate, and we're testing this. We don't know for sure yet. And we believe that because in the plasma of mice there's a carboxylesterase enzyme which rapidly converts ethyl pyruvate into pyruvate. Gut bacteria also probably converting ethyl pyruvate into pyruvate. And so the form the ganglion cells is probably seeing is pyruvate, but this does stabilize it in our drinking water. However, we are interested if any of this is making it to the eyes ethyl pyruvate, and so the studies are underway, because if it's more cell penetrant or more mitochondrial penetrant, that could explain improved effects as well. And for human treatment, that could be important. Would this work much better than pyruvate? Though more work is needed to prove all of that, there's a reasonable chance that it would. But in humans, we might need different concentrations to the mice and possibly much lower concentrations if it's not being converted to pyruvate in the blood or gut. So we tested ethyl pyruvate, and here you see experiments with ethyl pyruvate, and indeed it has a very nice rescuing effect. Here you see the wild type untreated. Here you see the untreated mutant again getting the deep chambers. And you see the pyruvate treated where we lower the anterior chamber deepening. Not all of them have no chamber deepening because some of them get higher P. This house has a mild chamber deepening. And if you look at the chamber depths here, again, you can see there's a clear difference between the pyruvate treated and the untreated mutants. If we look at intraocular pressures, again, we can see this strong protection from intraocular pressure. And if we look at optic nerve damage this time, we are preserving optic nerves. There's a low incidence of no glaucoma in black here, and a low incidence of the most severe group with some more moderates. Now, I want to point out that this is in a very severe genetic model of glaucoma. These mice get a very robust, they've got a very, very strong genetic risk of glaucoma, and it's early and it's severe. So having this protection here is quite a strong result because it's very hard to stop this particular glaucoma. So we're very excited by this and hope to progress, and we're testing combination doses because combinations of resilience factors may maintain peak efficacy, but at lower doses, which would be safer. So this is a goal of ours to optimize resilience factor treatments to maintain peak efficacy at lower doses. Why? Well, we had one rare severe adverse event at Columbia with nicotinamide at 3 grams per day. This affected liver function. When the person went off the liver function, it was reversible. But it does mean you need liver function tests and need to be careful if you're using 3 grams per day. Even if that is a rare result, we still have to be careful. So ideally, we'd use lower levels. It's possible that in some populations, people might be intolerant to nicotinamide or pyruvate, gastric upset or other instances, so they won't tolerate it. Again, lower doses, if we can combine molecules, would be better for patients. A recent study suggested increased cardiovascular risk by using nicotinamide levels. I want to point out this is controversial, not everyone accepts it. And I don't personally know if I believe it's that strong a risk yet, but being as it's been suggested, we should be careful. And just because I'm not yet convinced doesn't mean we don't have to take that care. So if we can lower the doses, that's a good thing. If we can keep peak efficacy, there's no arguments that I think that would be a good thing to do. So we're actually combining resilience factors to allow the maintenance of peak efficacy, and that's an important goal of our research going ahead. So very simply, our goal is if we're using single higher doses, we have high efficacy, but we're getting some intolerance and some risk of severe events. By combining them at lower doses, especially lower nicotinamide, we're trying to achieve a maintenance of high efficacy, but with greater tolerance and a very low risk of any really severe adverse events, maybe completely negligible. So we've got some initial data in this very severe V265D LMX1B glaucoma. Now, these aren't doses as low as we'd like to achieve for the future, but they're a start. Initially, when we used nicotinamide and prove it, we've used doses equivalent to about 20 grams per day, such as that 10 grams per day, such as that previous slide I just showed you in the LMX1B model with pyruvate. So we're now continuing and we're combining our 3 grams of each equivalent dose to see what happens and to see, can we make it as effective as the 10 grams per day of either was? And indeed, our initial results are very promising. And combined doses are more effective against intraocular pressure elevation, as you see here. Here's the untreated mutant, nicotinamide alone, pyruvate alone. The combined is clearly more protected. And excuse me, if you look at nerve damage scores, this is initial data, but it's still highly significant right now. And you can see the nicotinamide's protective, less with glaucoma, the pyruvate's protective, and the combined was more protective and much more highly significantly so. So this combined lower dose is as effective as our original very high dose. So that's very encouraging. And we're now hoping to continue and test even lower doses to see if we can make these differences in the drainage tissues and ultimately the nerve. We've also been looking at doses and their effects on cell death caused by this LMX1B mutation. And as you can see, we can have the untreated or the treated, and if we do tunnel assays for apodosis in the trabecular meshwork, we can increase the rate of apoptotic cells in the trabecular meshwork, and we can actually restore morphology by resetting development in a more healthy way. So if we take a P21 mouse with the LMX1B V265D, here's a region of Schlemm's canal which is clearly very atrophic, didn't develop normally, or maybe some cell death. If we take an animal that had the combined treatment, the Schlemm's canal is in much better shape. If we look at the trabecular meshwork, a similar story. I should mention that these images are from flat mounts of the anterior chamber of the angle region, where we can analyze whole quadrants or whole limbus of the eye and make 3D models and reconstruct them. Here's a model of a constructed quadrant of a mutant, where you can see there's lots of missing tissue and the tissue looks pretty sick. Here's an example of a combined treatment, which is much more robust, and we're quantifying this, and it is significant. I'm sorry, I don't have that on the slide. So we're finding that these treatments are lessening trabecular meshwork cell death, and they're lessening developmental anomalies. So this is why I mentioned early on, there's potential promise here, even for resetting development in childhood glaucomas. And indeed, there's an extended postnatal window of development of the angle in mice, which appears to be true in humans as well. Although we don't have exact timings of how long this would be effective, maybe we could treat out to a year or maybe further, and time will tell. But if we could make that difference in the early stages of life using relatively benign and nutrient-based molecules, that could be remarkably helpful in preserving the health of the angle for life. So key messages to end. I've shown you that metabolism and key messages about metabolism and glaucoma vulnerability. I showed you age-dependent decreases of NAD, which increase the likelihood of metabolic decline and contribute to glaucoma. I showed you the nicotinamide treatment is potently protective. I also showed you disturbed glycolysis and pyruvate metabolism contribute to the progression, and that pyruvate administration is also protective. These metabolism preserving treatments have been highly reproducible in large numbers of animals, and so they're robust. The LMX1B mutation perturbs metabolism in the drainage tissues, and NAM and pyruvate treatments also lessen early onset IOP elevation in mice with a very high genetic risk of glaucoma, and can also lessen developmental abnormalities in the angle. So metabolism supporting treatments have great potential for glaucoma, and I surmise that combination treatments are likely to be the most effective and offer the possibility of lower doses, even though there's significant work remaining to be done. Relevant to human glaucoma, there's an emerging and growing literature supporting mitochondrial susceptibility and altered metabolism in the human disease. There's increased mitochondrial DNA mutations in POG, the affected controls. Importantly, these mutations occur in blood cells, suggesting systemic susceptibility. Also now, individuals with lower mitochondrial function have more rapid progression to glaucoma. This again was assessed in blood cells, a very important paper in Nature Medicine by Ted Garraway-Heath and Vladdy Petriti last year. Nicotinoid levels are also low in the blood of POG patients, and pyruvate levels have been inversely associated with severity. And finally, pyruvate and age-related metabolites are resilience factors against human glaucoma. This is very clear from studying the UK Biobank cohort, with pyruvate, lactate, and citrate conferring resilience to a high genetic risk of glaucoma. These factors at their higher levels offer protection with 29% less glaucoma at the highest PRS if you have higher levels of these molecules. And so both polygenic and metabolic risk should be considered in looking at glaucoma and predicting risk. Clinical trials are underway to test the benefits of nicotinamide or nicotinamide with pyruvate administration, and they've got initial promising outcomes. So I'll stop there. Just along the members of my lab, Nicholas Tolman was very important in what I showed you today, as well as Ravati Balasubramanian and Pete Williams and others. And thanks also to our collaborators. And I should mention, I didn't show the data. We've also been collaborating with Brian Jones and Rebecca on these metabolic studies, and they've been generating the most remarkably beautiful EM of large segments of the trabecular meshwork, which is important in our metabolic studies as we proceed. I'm sorry, Brian, the data isn't all analyzed yet, so I didn't show it today. I forgot to put you on the slide. I apologize. Thank you.